All right, well, as Dr. Gibson said, I'm going to talk today about a field that's been near and dear to me for decades. When I was an undergrad, I started working in a herbarium, and it was mind-blowing. Other people walk into a herbarium and are horrified. So it certainly takes different personalities, but by the end of today, to appreciate, but by the end of today, I hope that you all appreciate the diverse utility of a herbarium if you have never even set foot in one yet. SIU has a herbarium. A university this size, we have quite a large herbarium. I'll talk more about the specifics. Before I get there, as part of Darwin Week, I hope many of y'all attended our Darwin Week activities. We have an art show and a display for the herbarium on Art Alley. So there are 3 cases. Go check it out. In particular, there's one case with an explanation of the herbarium, its ties to Darwin, the history of natural history collections, their utility, and a spread of SIU herbarium specimens of different kind of import. It might be a cool thing in class, like you could have a little bonus activity, send students out there to go learn about the herbarium. I don't know, something fun like that. So what is a herbarium? I've had many of you in classes before, and I've explained all this ad nauseum. If you've taken my curation of collections class, but I still have to address this. Herbaria, plural of herbarium, they're research and education facilities. They house dead, dried specimens, and to the right is a nice example of a dicanthelium, which somehow Dr. Gibson just sensed I would talk about. This is from our collection. It's got all the salient features here. Let's see if, yes. We have a label to the bottom right. We have the physical plant specimen. We have a barcode, which is brand new. And we have this historical identifier for a particular specimen called an accession. So it's got an accession seal and an accession number. This is problematic in our collection, these accessions, because they're duplicated and misnumbered and very complex. Our past curators did a confusing job of adding accession numbers. Then, this is actually a photo. I know that's shocking. This is a photo taken as part of our imaging process of specimens, which I'm going to talk about momentarily. It's got a color guide, and it's got a ruler for scale. And so one of our goals is creating high-definition images. Herbaria, there are far more than 3,100 worldwide, but these are just the registered real herbaria. There's a lot of private, more than this when you account for the private collections. And there's estimated to be at least 390 million herbarium specimens just like this worldwide. Here's a map of all those different institutions worldwide. There's a ton in North America, as you can see, and we're one of those little dots. Herbaria are houses for specimens. What kind of specimens are there? There are vouchers for studies. There are casual collections that, hobbyists or students or faculty or just people go out, collect specimens, they find them of value, they press them, they put them in the collection. But the ones that are super valuable are the ones that are vouchered in a floristic study in Southern Illinois. And those are really rich components of our collection. Past graduate students, especially at SIU in our program, especially in the 50s, 60s, 70s, and 80s, were doing floristic studies in little micro areas throughout our area, especially in Southern Illinois. And they made vouchers of all the species that they found. So that's a huge, rich part of our collection. And as a consequence, we have a lot of Southern Illinois plants. The data that are on these labels are quite diverse, but this is kind of typical for our collection. And it's got a name, it's got some brief locality information. Sometimes there are coordinates or there are township range and section information. It has a date, so we know when it was collected and what year. And so it's a frame of reference of where, when, what, and sometimes how or why. And as scientists, those are fundamentally all the questions we ask, right? And those data are here. I'm going to circle back to that at the end. These are ideal resources for identification. So at the very least, herbaria are super useful for plant identification. Our herbarium has 90,000 specimens, we think. We're still figuring that out. Some collections from around the world, I've kind of broken it down by geographic area here. Starting at noon, we've got all the Illinois and Missouri collections, southeastern states, the rest of the U.S., and essentially the rest of the world. So quite clearly, we have a ton of Illinois specimens, especially from the southern portion of Illinois. And we have, when you kind of combine that with the southeastern flora, it's well over half of our collection is included into that kind of category. And we're unique. So specimens, you go to a tree, right? Clip a plant, a bit of the branch off, press it. Well, for that tree, you could clip hundreds of little branches and press them all and glue them to sheets of paper. Those would be duplicates. And the more it's duplicated, maybe the less unique it is. And we have lots of things that are not duplicated anywhere else. They are unique collections. So for decades, SIU, long before, see if y'all get distracted by the video. Is immediately what I thought of when Dr. Gibson started talking about bad slides. For decades, the collection has been digitized in terms of transcription. So label data has been transcribed into databases of varying quality up until they were that database was kind of mothballed. Dr. Garwood came in. This is Dr. Nancy Garwood, if you've never met her. We both curate the collection of the SIU Herbarium. And you came in 2009, right? No, 2000.
That's when I started a herbarium.
Yes.
I got here a little early.
You want to take your time. You don't want to. 2009, she pulled that database out of mothballs and she revamped it, fixed it, and started in mass increasing the digitization because it was left off less than halfway through the collection. In 2019, 10 years later, we finished, which means we were just systematically going through cabinets, transcribing the label data from all these 90,000 collections. And this was like a really important milestone for us. We were waiting for this milestone so that we could seek funding. And that's kind of the whole import of today's seminar, is that we got money and we're doing a lot with it. And we hope that you will do something with it soon. So our herbarium is here in this little star. This is kind of a plant community in the broad scale kind of map of areas, different community types. Here's another map that shows many of the regional herbaria throughout the southeast. I tried to make an argument in a previous slide that our holdings are very strong for southeastern flora, and we had a very similar, we felt like we had the best tie to other collections in the southeast, because geography matters in terms of seeking funding for collection management. So, since it's so strong in the southeast, we wrote to join a TCN. A TCN is a thematic collections network funded by this program that we're applying to. We said we want to join this group of herbaria to digitize our collection so that all of this information for the southeastern flora would be even richer. Now we're in this unique position of being right at the corner of the southeastern flora in many ways. And so it's a really interesting distributional space of plants for much of that flora. This is kind of the extent for a lot of those taxa that are found throughout the southeast. We wrote a proposal and we essentially joined this consortium known as CERNAC. And if you've never looked online at CERNAC, it's a really valuable resource. I'll show you what the website looks like in just a second. It compiles all the herbarium specimen data across essentially all of the U.S., but you can restrict it just to the southeastern flora. So we're digitizing. I've talked about transcription as an aspect of digitization, where we literally just type in label data. But the whole specimen is very rich in information. There's all kinds of resources that can be used from that for scientific data. And it's not just us, it's not just in the U.S. This is really a worldwide effort in order to make all these historical collections available for scientists anywhere. There's been a really big problem in the past of things being in very inaccessible places and hindering our understanding of biodiversity simply by not being able to access those specimens. And with this, we hope we kind of forego many of those problems or overcome. So 3 major components to digitization. There's a lot more nuances and complexity here, but I'm just going to break it down to our major initiatives here. One is to image specimens, one is to georeference specimens, and one is to transcribe and barcode label data. So just to make sure we're all on the same page about what digitization means. CERNAC, which I mentioned just a moment ago, this is their website. It's quite nice. It's kind of basic, but it's also really useful. It's not like Google, not that sophisticated in terms of reading your mind. Just as you typed in one letter and it knows exactly what you're thinking and it fills in the rest. No, it's not quite like that. But it does do a great job of effectively searching for things that you're looking for. So if there are collection data for whatever kind of field you want, who is the collector? When was it collected? In what state? In what county? What is the taxon? You name it, you can search by those fields and find everything that's known and digitized, which is amazing. This is an aggregation of data, but it's an incomplete aggregation of data because this bit is focused on just herbaria around the world, or specifically in the southeastern U.S. You might be interested in more than that. You might be interested in plants or animals or fungi found anywhere in the world. This is a program that's even more overarching than our TCN. It's called iDigBio. iDigBio is a data aggregator service and educational kind of program. It's a multi-million-dollar grant to essentially pull together all collection data of anything at any U.S. institution. It's incomplete, but hell, across all kinds of organisms, there's over 129 million records already. That's a lot of data. Really cool. And of course, we're digitizing. I'm not really doing the work. I have PI collaborators, Dr. Nancy Garwood and Dr. Jennifer Weber. We have grad assistants. They're both going to speak in just a moment. Janice and Breanna. And of course, we've had many undergraduate assistants over the years, and right now we have 8 working actively in the herbarium. So we're very productive this semester. We're doing really well, and again, I'm using this we like I'm doing most of the work, but I am. Let's just say what's really happening. I'm really important. I'm doing so much. You know who you are. I'm not going to list all the names. Sorry. So let's go through our 3 major activities of digitization briefly. So our first one, and they're kind of in order of how you can proceed if you're trying to do this. 90,000 is both like a lot, but seems like not that much, you know. But the amount of detail that's required in order to reconcile 150 50-ish years of chaotic management of things, physical objects, is daunting. Because our herbarium has existed since basically the inception of the university. When George Hazen French joined the faculty at SIU, he set up the first herbarium. Most of it was his own collections. And at that time, it was probably only a few thousand specimens. But imagine dozens of curators over the years with different curatorial activities, different thoughts on what's important. There were no computers in the 1850s, obviously, and 60s. We're left with a vestige of complex history of what has happened to these specimens in that time period. We have big steel cabinets where these specimens are held, and as we process specimens, we pull them out. We bring them to a computer, and they get typed into a database to parse all the information that's on that label. It's kind of simple, but it also kind of takes a human brain to parse those data accurately. So for example, this one sentence here, sandy ground at the Blue Spring camp, has several different aspects in it. It has both like soil type and a specific name, place name. It's not really one sentence when you think about parsing fields. These would get separated into different fields describing the different aspects. And that's kind of hard still for a computer to do, but relatively easy for a human brain to figure out. Much of this seems kind of silly and inconsequential, and if you only had a database of a single specimen, you couldn't do much with it. But with 90,000 specimens or hundreds of millions of specimens that are aggregated across the world, you can do amazingly powerful things. So we especially want to know the scientific name, the date it's collected, and where it was. This is literally who, when, and where. Because you can ask a lot of cool questions with that. And we are imaging specimens. So as I showed you this image earlier, here's our imaging setup. This is Mary right here taking photos with our copy stand. There's a little SLR camera right here, 2 light diffusers to create a very omnidirectional kind of diffuse light source so there aren't a whole lot of shadows. And we have a nice computer to visualize all this with a setup where bling, 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 take a photo. You can label the file of that photo with a barcode reader because we've already put barcodes on, and then we can confidently link database with image data immediately, which is really important. Because as I said, it's super easy for things to get chaotically confused within a collection. Today we just hit 5,000 specimens imaged, which I know I'm excited. And we've just started imaging like 3 weeks ago-ish, so we're making really good progress. This isn't the big bottleneck. Our transcription phase is our big bottleneck, and we are currently at 26,000 specimens on our transcription phase. Our last phase, which we haven't started yet, is georeferencing, converting either township range section information into coordinates or converting words, descriptive words of an area into coordinates. This is known as georeferencing. Because most specimens are old, especially in our collection, most specimens were collected before GPS existed. And so what we have are a real mix of locality information. Fortunately, there are softwares like this, Geolocate, which we can use to help estimate. And some things are just hard and we have to do it more manually. Some things can be semi-automated. And it's one of those things where you have to have all the data to kind of efficiently do it. All the kind of label data ahead of time to kind of parse it, figure out how you're going to treat these. So this is our 3 main phases of digitization of what we plan to do. We're at various stages of that. And once we're a little bit further, One of our project goals is to look at phenology variants over time across taxa to see how our spring flora has been affected in southern Illinois. And what I mean by that is, by phenology, is we have seasonal life history traits that are variable. You know, when do the claytonias, for example, flower in the 1920s versus 2020? There may be differences. We can not only look at this in terms of our collections, but as I said, we have these massive national databases where we can aggregate data. There are other Illinois collections that aren't just here, and we can code based on phenological state and understand processes like climate change by using these data. One of our other aspects of what we'll be doing in terms of how the herbarium is interacting with the public is through outreach activities. And Breanna has already undertaken some outreach activities, and I'll let her take over, and I'm going to come back in just a moment.
So many of you would probably agree that outreach is a really important component of any scientific endeavor, because when we're doing our projects, we should be thinking about how are we going to portray this to the public, and how can we potentially connect them to our project and get them involved. And since 2015, every summer I've been working at Wren Lake doing scientific education, environmental education, and outreach. So I was really excited that for this project, because it's an NSF-funded grant, there's an outreach component that has to be enacted. So I got to do that, and it was really exciting. And many biologists don't know what herbaria are, so a lot of people in the public don't know what herbaria are, or even natural history collections and what kind of data is available. So that was kind of the overarching theme and goal of this outreach activity. So I'm going to play this YouTube video, and it gives a nice overview of what we did. Grant, the U.S. Army Corps of Engineers at Wren Lake hosted the outreach program, Build Your Own Botanical Collection, presented by Breanna Whitley, a plant biology master's student at SIU. The goal of this program was to engage an informal audience about herbaria, the importance of natural history collections, and the use of herbarium specimens. This outreach event attracted a diverse set of participants, with individuals ranging from the pre-K level to even retirement age. There were people with experience as master naturalists and also people with very limited botanical knowledge. During the program, participants learned about natural history collections, with an emphasis on botanical collections. Then there was a demonstration about how researchers collect plants, followed by proper pressing techniques. To make the program even more accessible to the general public, there was also a demonstration on how to collect and press plants using household items. All of the 60 participants received a mini homemade plant press to test out their newly learned skills. The indoor presentation was followed by a field trip to the Wren Lake Pollinator Garden, where participants were encouraged to collect plants to create their own botanical collection. As a reminder, no person is permitted to collect plants on public lands without prior permission and documentation. Build Your Own Botanical Collection is an outreach program provided by staff and volunteers of the SIU Herbarium. If you'd like to incorporate this program into your curriculum or special programming, please contact us. Cool. So yeah, again, that's an overview of the activities that we did. And as it mentioned, we started indoors, and I gave a brief introduction about natural history collection. And I put it in the frame of reference of museums because it's something that people are familiar with. So that was a way to kind of relate to the general public. And then the nice part, and the part that I found most fun, was that we made individual plant presses for all of the participants. So they actually got to go out and collect plants, press the plants, and then think about how they might use them once those plants were dried. So if you know any organizations or agencies that would like to have this type of programming, let us know. I'd love to do it again this summer, and I'm hoping to do it at Wren Lake and maybe also some other agencies in the community. So if that's something you might be interested in, just let us know.
Thanks, Breanna. Incidentally, fancy camera work, right? Thank you. If you haven't yet checked out the School of Biological Sciences YouTube webpage, we have many herbarium tutorials, and we have a lot of other content up that you might want to check out. So now, since I kind of set up that, the herbarium data are an incredible resource for research in a lot of ways, Janice would like to talk about some aspects that you can use herbarium data to address some cool scientific questions.
Thank you. Yes, I want to share with you a project that I was involved a year before coming here to U.S., and it was this project about surveying orchids in Colombia. As we didn't have many of our collection digitized, and few of them even have images, we had to do everything kind of by hand, go to the herbaria and study them there. So this is Colombia. I imagine that most of you know that we are kind of in this corner of South America. This is how the country looks like, and basically when you see it, you immediately kind of identify a big cordillera in the middle, that is the Andes, and then we have kind of 5 regions using that cordillera in the middle. So if you imagine the cordillera is one region, and then you go to the west and you have that long piece that is flat, that is the Chocó region, that is super rainy with the rainiest place in the world. And then you go to the north and you have kind of a yellow area, and that's a dry forest. And dry forests are so cool because they lose their leaves when it's dry, but they keep fruits and flowers. But when it's wet, they keep all the leaves, but you can't find fruits and flowers. So you have to go and collect them twice if you want to have everything together. Okay, and if you go to the east, you have this kind of yellow area, that's the Llanos, that is just plain savannas, just grasses. And then in the south, all of this green is the Amazon forest, the biggest rainforest in the world. And an interesting thing is most of our orchids are epiphytes like this, like in most tropical regions, they live on trees. And depending on this, then you kind of expect to have more orchids in some regions and less orchids in some other ones. For example, we did not expect to have many orchids in plains, in the Llanos, and we expected to have more orchids when we have trees, like in the Andes or in the Chocó region. So this is more or less what I was mentioning before. So if we go to the Chocó region, we have this beautiful forest. When you have epiphytes, and epiphytes growing on top of epiphytes, and on top of that you have bryophytes and lichens, and everything is just growing on top of each other. It's super humid. And then you have the Andes that have all of these different climates. When you go to the mountains, you grow in elevation and you have different type of ecosystems. And then you have the Amazon forest, and this picture is specifically kind of ecosystem in the Amazon forest because it's not just forest. There are some areas that get flooded like this, the Igapos forest, and the water just goes up and up and up, and you can basically take a boat through the canopy, and you go basically on the canopy with a boat. So you can imagine that in some of those forests you can't find much epiphytes, right? And then you go to the Llanos, and the Llanos is those beautiful savannas that I mentioned, and you go to the north, and you have these deserts, cacti, and the dry forest that I just mentioned. So using all of this information, we start searching for orchids, and our first source was, of course, herbaria. So our purpose in that study was to create a big database with everything that we can put together. So we put together information from herbaria. We had more than 15 herbaria that collaborated with us. Some of those herbaria, as I mentioned, didn't have the information digitized. We got some databases. For example, Missouri Botanical Garden sent us their database digitized, but most in Colombia didn't have, so we had to go to the herbarium. We had students that were there days and days and days digitizing information. We had also a list of orchids that somebody made. Some specialists said, I think this is what it is in Colombia, and we have a list, but we didn't have that list associated with the record. So it was not possible to go and check if that something changed the name or something was rediscovered, or, it was not possible to go back and see if that name is, still valid or not. But we have those. And we have floras and some taxonomic treatments that we also put every single specimen that was mentioned in those documents out, and we digitize every single one from them. And then using that information, we create a new list with records and localities, and then we georeference as much as we can. We did it more or less for 60% of our data. Then this is what we found. We found that Colombia was the richest country in orchids in the world. We have more than 4,200 species. We found that 1,300 species were never recorded before. 30% of them just grow in Colombia. They are endemic of our country. This is how the georeference specimens look like. And this showed to us that the richest region in orchids is the Andes. But of course, also show how some researchers go and collect. So there are places that nobody goes to see, and then you can look at this and say, okay, we need to go and explore some areas that nobody has been before. And when we go to the Andes, we found out that most of our orchids grow kind of in the middle of that elevation. And this is more or less what I have here. When you go through the different elevations in the mountains, we have different ecosystems, and most orchids prefer kind of that middle range between the lower mountain and upper mountain forests. And we found out that some orchids live just in one little kind of elevation range. And so you have replacement of different group of species when you move up in the mountains. And if the mountains are wet in one side and dry in the other side, you have a replacement that is different in each side of the mountain. So all of that we can find out because we have all of this information. Finally, we found out that our national parks host very little of our species, less than 20%, and less than 14% of our endemics. If we go and check other protected areas, like private areas, they host more or less 1,000 species, which is still less than 50% of our orchids are protected in these areas. So we suggested that they should be increased and cover different spectrums of the whole elevation if we want to preserve most of our phylogenetic diversity. And finally, we couldn't make this possible without all the database that were shared with us and all the possibilities of going and visiting those herbaria and to be, go in there and check the specimens and get the information. All of this was done just with the specimens. We didn't go and collect anything. So we got answers, not just in taxonomic point of view, but we have also ecological questions that we can ask, biogeographical questions that we can ask, and also help in conservation and say, hey, we have tons of good species that need to be conserved. They are so special in this region. We should make a new reserve or a new protected area here or there. So this is what we did, and this is why herbaria are so useful. Thank you.
To put a little cap on that note, I think many people might hear this and think, oh, there's no use for herbaria in my interests or my research, but I know that herbaria can be useful in every field of plant biology. Physiologists use herbaria. Ecologists use herbaria. Taxonomists use herbaria. Everybody can use herbaria for their research or should be sharing vouchers of their study systems with herbaria to record things that they're studying for posterity. You're looking at a system, how the hell is anybody ever going to know what you meant when you said you have this plant without a voucher in a herbarium? Otherwise, the data that we're producing in the herbarium, there's going to be a lot of it. I mean, it's a lot of data. It's going to be free data. We have it. We will have it. We have some. We will have more. It's a huge opportunity. a lot of opportunity for future research or even future money. We have jobs. We have students now. The students graduate. Janice and Breanna are graduating. I need a new graduate assistant that's going to work in the herbarium. We've been advertising, but we don't have anybody yet. The student will be advised by Dr. Weber, and the student will work in the herbarium. Sure would be nice to have somebody. We don't have someone yet. And we're always looking for more undergraduates who can continue the digitization effort as we move forward. And I'll give acknowledgments. This was done by many, many students over the years, including our many current students. Funding by several NSF grants have helped the digitization process. We've had funding from the now extinct SIU UGA program. That was really wonderful when it existed. Sure wish it still existed. And none of this could be possible without the consortium help of the CERNAC, TCN, and IDIG Bio. So thank you all very much.